शीतला सस्यश्यामला मातरं वन्दे मातरम् शुभ्रज्योत्स्ना पुलकितयामिनी पुल्लकुसुमित द्रुमदलशोभिनी सुहासिनी सुमधुरभाषिणी सुखदां वरदां मातरं वन्दे मातरम् वन्दे मातरम्.
Good morning, students. How are you? I think you all are fine. Is it right? Yes. In the last class, we learned about cell is the basic unit of our life. We started that lesson. Cell is the basic unit of our life. So, in that, we learned so many issues. Who invented the microscope and also discovery of the cell. Is it right? So, who discovered the cell? Robert Hooke. He discovered the cell. In where? In oak tree bark. Is it right? So, what he find out? That cells are dead cells. That cells are dead cells. In a dead plant. Dead cells, dead plant. Because that is a bark. That is a bark. So, what he observed? That cells are dead. So, actually, when the Robert Hooke he find out the cells in a bark of oak tree, that was a milestone. See, the discovery of the cell by Robert Hooke was a milestone in the history of science. Yes. రాబర్ట్ హుక్ కనిపెట్టినటువంటి సెల్ ఏదైతే ఉందో ఆ సెల్ అనేది హిస్టరీలోనే ద సైన్స్ హిస్టరీలోనే ఒక మైల్ స్టోన్ లాంటిది. ఒక మైల్ స్టోన్ లాంటిది అంటే అంత ప్రాముఖ్యమైనది అనమాట. వాట్ హీ అబ్జర్వ్ దట్ సెల్స్ ఆర్ డాష్. వాట్ ఈస్ దట్? డెడ్ సెల్స్ మీన్స్ మృత కణాలు, జీవము లేనటువంటి కణాలు. దే ఆర్ కాల్డ్ డెడ్ సెల్స్. It means inside the cell empty, there is nothing inside the cell. No, nothing is there. So that's the reason. Nothing means inside the cell some components are there, but that all the components are absent. So that's the reason that cells are called as dead cells. What is that? Dead cells. Do you know what Robert Hooke did? That experiment we also did with the help of a matchstick. Is it right? We didn't get an oak tree. So that's the reason. Instead of oak tree bark, we took matchstick. With the help of matchstick, we did that experiment. What Robert Hooke did. Is it right? Robert Hooke ఏ ఎక్స్పెరిమెంట్ అయితే చేశారో ఓక్ ట్రీ ద్వారా అదే ఎక్స్పెరిమెంట్ మనం అగ్గిపుల్లను ఉపయోగించి చేసాం. ఇస్ ఇట్ రైట్? అగ్గిపుల్లలో కూడా అగ్గిపుల్ల అనేది ఇట్స్ నాట్ ఏ లివింగ్ ప్లాంట్, ఇట్స్ ఏ డెడ్ పీస్. మ్యాస్టిక్ మీన్స్ it is a dead part. So when we cut it and when we see the part under the microscope, what we observe that cells also dead cells. Okay na? Right. But is it possible to see living cells? Is it possible to see living cells? Yes. Is it possible? So living cells, how can we see? Living cells means Sajiva Kanal. What is that? Sajiva Kanal. It means inside the cell, everything we can able to see. We can able to see. Such kind of cells are called as living cells. So, how can we see living cells? How can we see living cells? For that, we have to do one activity. We have to do one activity. అంటే మనం బ్రతికున్న కణాలను కూడా చూడడానికి ఒక ప్రత్యేకమైన ప్రయోగాన్ని, ఒక యాక్టివిటీ, ఒక కృత్యాన్ని మనం చేయొచ్చు. వాట్ ఈస్ దట్ యాక్టివిటీ? వాట్ ఈస్ దట్ యాక్టివిటీ? సో for this experiment or for this activity, we need some of the operators. We need some of the operators. One onion. What is that? One onion and one sharp blade. One sharp blade and slide, and also microscope. And also microscope. Little amount of water also. Little amount of water. Onion, blade, slide, water and microscope. So these are the operators for doing this experiment. These are the operators for doing this experiment. First what we have to do? First we have to do. So take a onion and remove the peel. Remove the peel. So I am removing. See, remove the onion top layer. That is not useful for doing this experiment. See, in our Bhasha Telugu we have to say pora. Pai pora ne teesiveyala. What is this? Pai pora teesiveyala. Okay na? So like this we have to remove the top layer first. Okay now, right. So like this we have to remove the top layer. Then after that there is a thick layer, whitish or pink colored thick layer we have to observe. So that layer we have to take out first with the blade. With the help of blade. See, with the help of blade I am taking one thick layer. See, like this. See how I am taking? Like that. Okay now? So after that carefully we have to fold it. Fold it. Yes. See, I fold it. It cut it as a two pieces, but when it separates, the under the layer there is a thin layer, white colored thin layer is there. See, carefully observe. Yes, see. Yes, this is the thin layer I take out from the onion peel. Is it right? So this layer we have to keep it on the slide. We have to keep it on the slide. See, like that. We have to keep it on the slide. Like that. Then pour 1 or 2 drops of water. Drops of water, like that. See? Yes. So now this slide we have to keep it under the microscope. We have to keep it under the microscope. Yes. I kept under the microscope. You think like that. You imagine. I kept under the microscope. Then I have to observe. When we observe this slide with the help of microscope, there are some cells appear to our naked eye. So that cells, how it is? See, look at the board. The cells look like a rectangular in shape. A rectangular in shape. See how the cells are? Rectangular in shape. These are the onion peel cells. These are the onion peel cells. So actually, these all the cells are living cells. These all the cells are living cells. Inside some organelles are there. Some liquid material also is there. Everything is there in the onion peel cell. So that's the reason these cells are called as living cells. But what Robert Hooke observed, in that cell, we observed empty space. In matchstick, we observed empty space. Robert Hooke, he observed in a oak tree bark, he observed empty space. Because that cells are called dead cells. But these cells are called living cells. Living cells means inside some fluid-like material is there, as well as some of the components also is there. They are called cell organelles. What is that? Cell organelles. So inside the cell, some components and also liquid material. So one who is having this kind of organelles and liquid material, that cells are called as living cells. Sajiva kanalu. What we call Sajiva kanalu. అంటే వాట్ వి అబ్జర్వ్ నౌ, ప్లాంట్ లో ఉండేటువంటి లివింగ్ సెల్ చూసాం. ఇప్పుడు మనం సజీవ కణాలను చూసాం. ఆనియన్ అంటే ప్లాంటే కదా అది. ఎస్. సో ఆనియన్ ప్లాంట్ కాబట్టి దిస్ సెల్స్ ఆర్ బిలాంగ్స్ టు ప్లాంట్ లివింగ్ సెల్. బట్ వాట్ Robert Hooke observed, oak tree bark, that is also plant, but he observed dead cells. He observed dead cells. Now what we observed, these are called living cells. Both are plants only, but in the plants some of the dead cells also is there, dead cells are there, as well as living cells also is there. This is the way to see the living cells in onion peel. In onion peel. Okay now, right. This is about onion peel cell experiment. Okay. Next, if you want to see animal living cell, what is that? Animal living cell. For that, what do we have to do? Shall we go for animals? No need. We are also social animal, is it right? From our body, we have to take little amount of tissue. With that tissue, if you observe through the microscope, we will find out some of the cells. That cells are called living cells, because we are living body. No? Our body is living body, so that's the reason. That cells are also called living cells. How we will see? First, from our mouth. Actually, what is this experiment? How to see the 6 cells? 6 cells means bugga kanal. What is that? బుగ్గలో ఉండేటువంటి కణాలను మనం సెల్స్ని ఏ విధంగా చూడవచ్చు. Is it possible? Yes, it's possible. So, first what we have to do, before doing this experiment, ప్రతి ఒక్కరు కూడా మొక్కని నోటిని శుభ్రంగా కడుక్కోవాలి. ఈ ఎక్స్పెరిమెంట్ చేయడానికి మనకి ఒక వుడెన్ స్టిక్ ఆర్ వుడెన్ స్పూన్, ఐస్ క్రీమ్ తిన్నప్పుడు స్పూన్ తీసుకుంటాం కదా, దట్ స్పూన్ వీ హ్యావ్ టు యూజ్ ఫర్ డూయింగ్ దిస్ ఎక్స్పెరిమెంట్. అండ్ స్లైడ్, వాటర్, microscope. These all are common for doing this experiment. So first, before doing this experiment, we have to wash your mouth. Then take a wooden stick. But I don't have a wooden stick, but instead of that I am using this. What is this? Matchstick. Then open your mouth and put this wooden stick into your mouth and scratch it. Then little amount of tissue will come. Don't do hard because it will damage. So that's the reason. lightly you have to scratch it on your cheek. Then little amount of tissue will come out. Then that tissue we have to take and keep it on the slide. Keep it on the slide. Yes. Then put a water drop on the object. Then this slide keep it under the microscope. If you observe, the cells appear like this. The cells. There is no specific shape. The animal cell, they don't have specific shape. But if you see the plant cell, the cells are in rectangular in shape. Okay, na? What we see? We saw the plant living cell and animal living cell. There is a major difference between plant cell and animal cell. That is cell wall. In the plant cell, there is a cell wall. Whereas in the animal cell, the cell wall is absent. Okay. A significant observation. A significant observation. It means ఒక కీలకమైన పరిశీలన. కీలకమైన పరిశీలన. Who did it? ఎవరు చేశారు? Robert Brown. Who is that? Robert Brown. What is that? What is that? He find out a nucleus in different cells. He find out nucleus in a different cells. Because of this information, we are clearly understanding about cell. Because of his investigation or because of his observation, nowadays we are very clearly understanding about cell. It means Robert Brown, you find out the nucleus in different cells. You find out the nucleus in different cells. But before that, before the Robert Brown, Felicina Fontana and other scientists, Felicina Fontana మరియు ఇతర శాస్త్రవేత్తలు, the first they saw the nucleus in epithelial cells in the year of 1730 to 1805. Felicina Fontana and some other scientists, they first saw the nucleus in epithelial cells. Epithelial cells means epi means paina. అంటే జంతు చర్మములో, అంటే జంతువు యొక్క బాహ్య చర్మం, అంటే మన బాడీ మీద పైన ఉండేటి చర్మాన్ని బాహ్య చర్మం. That is called in English we have to say epithelial cells. In the scientists means the Felicina Fontana and other scientists, they first saw the nucleus in epithelial cells. బాహ్య చర్మంలో ఉండే కణాల్లో they found the nucleus. They found the nucleus. But they found only this nucleus in epithelial cell. But they did not found this nucleus in other cells. They did not found this nucleus is in other cells. That was made by Robert Brown. See? But nucleus in different kind of cells, and recognizing that was an integral part of the cell goes to Robert Brown. What is this meaning? ఈ సైంటిస్టులు ఓన్లీ ఎపిథీలియల్ సెల్లోనే న్యూక్లియస్ చూశారు. బట్ దే డిడ్ నాట్ సీ దిస్ న్యూక్లియస్ ఈజ్ ఇన్ అదర్ సెల్స్. దే డిడ్ంట్ సే. బట్ హూ టోల్డ్ us? That is Robert Brown. Robert Brown, this nucleus we have to see in different kinds of cells. We have to see in different kinds of cells. Not only that, this nucleus is the integral part of the cell. What he said? This nucleus is the integral part of the cell. అంటే కణములో ఒక అంతర్భాగమై ఉంటుంది అనమాట న్యూక్లియస్ అనేది అని చెప్పాడు అనమాట. ఏంటి న్యూక్లియస్ అనేది చాలా రకాల కణాల్లో ఉంటుంది అండ్ ఆల్సో ఇది కణములో ఒక ఇంటిగ్రల్ పార్ట్, అంటే కణములో ఇది అంతర్భాగం, అంటే కణములో ఉంటుంది కంపల్సరిగా అని చెప్పిన ఘనత రాబర్ట్ బ్రౌన్ అనమాట. వాట్ దిస్ సైంటిస్ట్ తెలిసిన పాయింట్ అదర్ సైంటిస్ట్ వాట్ చేస్తే దే ఫౌండ్ ఓన్లీ దిస్ nucleus structure in epithelial cells, but they didn't say anything about the nucleus. They didn't say anything about the nucleus. But this person, who's that? Robert Brown, nucleus he find out, and also nucleus find out in the cell means the same structure we have to see in other cells also. Not only that, this is the integral part of the cell. This information is given by the Robert Brown. So that's the result. Robert Brown is the first scientist, one who explained about nucleus. Robert Brown, where did he observe this nucleus? Where did he observe? He observed this nucleus in epidermis of orchid leaves. అంటే ఆర్చిడ్ అనేటువంటి పత్రం యొక్క బాహ్య చర్మంలో, బాహ్య చర్మం అంటే మీన్స్ పైన ఉండే చర్మం. ఇన్ ఇంగ్లీష్ వీ హ్యావ్ టు సే ఎపిడర్మిస్. ఎపిడర్మిస్ ఆఫ్ ద ఆర్చిడ్ లీవ్స్. ఆ చర్మంలో ఉండే కణాల్లో in a nucleus. How he find out what is the shape of this nucleus? What did he say? It is a circular in shape. It is a circular in spot. It is a circular spot. Circular in shape. Yes. And also, actually, when the Robert Hooke find the nucleus, from that day to the founding, the nucleus discovered by the Robert Brown. Do you know in between the gap means Robert Hooke to Robert Brown. Robert Hooke, what did he find out? Cell. From the cell to find out the nucleus, in between the gap 180 years. 180 సంవత్సరాలు పట్టిందన్నమాట. సెల్ కనిపెట్టిన తర్వాత న్యూక్లియస్ కనుగొనడానికి ఇంచుమించు 180 years. Okay. When the Robert Hooke observed the nucleus in epidermis of the orchid leaves, he found other cells also. Do you know what is that? Stomata. What is that? Stomata. You find out the stomata also during that time. What is stomata? Already you learned in the 7th class, I think. 7th or 6th. Stomata means it's a pores-like structures. Patra randralu. In Telugu, what we have to say? Patra randralu. And patram yokka epidermis lo undeటువంటి randralu. Patra randralu. You find out. Stomata is also found out by Robert Brown. What is the use of the stomata? The stomatas are useful for gases exchanging. అంటే ఏంటి? గాలి లోపలికి వెళ్ళడానికి, ఎట్ ద సేమ్ టైం లోన గాలి బయటికి వెళ్ళడానికి. గ్యాసెస్ ఎక్స్చేంజ్ జరగాలంటే మొక్కలో స్టొమాటస్ కావాలి. ద స్టొమాటస్ ఆల్సో ఫైండ్ అవుట్ బై రాబర్ట్ బ్రౌన్. What is the great significance by Robert Brown? That is find out the nucleus as well as you find out the stomata also. So it is a significant investigation, a significant observation by the Robert Brown. Because of this observation. Nowadays, we are very clearly understanding about cell. Very clearly understanding about cell. Okay, now next. Now, observation of the nucleus in onion peel cell. Already we saw how to see the onion peel cell. How to see the onion peel cell. Is it right? First we have to take onion. We have to take onion. Then remove the top layer. Then next very thick layer will come. From that layer, once little amount of piece we have to take out. Then fold it very carefully. And when we fold it, in between the folding there is a thin layer. That layer we have to take it and keep it on the slide. Is it right? Then when we see this layer with the help of microscope, we will observing some of the cells. Already I explained. But in the same manner, we have to do the same experiment again. Then so, if you see the cells of onion, inside the cell, there is a dot-like structure. There is a dot-like structure. Do you know what is this? Nucleus. What is this? Nucleus. How can we see this nucleus? The same experiment. So, when we observe these cells under the microscope, we have to see a circular and dark colored dot-like structure. When we find out, that structure is called nucleus. Okay now? So, if you observe animal cell nucleus, same experiment. Take a toothpick. Before that doing this experiment, we have to wash it our mouth. Then with the help of this toothpick, we have to scratch it. Little amount of tissue will come. The tissue we have to keep it on the slide. Then we will take one water drop and keep it on the slide. And this slide keep it under the microscope. When we observe, so the cells appear like this. See, here there is a specific shape, rectangular in shape, but here there is no specific shape. There is no specific shape. Here, both cells are having nucleus. Is it right? Yes. But there is a great difference between the arrangement of the nucleus in the plant cell and the animal cell. The nucleus position is different from the animal cell to plant cell, or animal cell to plant cell to animal cell. How? If you observe this nucleus in the plant cell, it's nearly cell membrane. It nearly arranged in the cell membrane. But if you observe in animal cell, this is the center of the cell membrane. What is that? The nucleus is arranged in center of the cell. అంటే ఏంటంటే ప్లాంట్ సెల్లో ఇది ప్లాంట్ సెల్, న్యూక్లియస్ సెల్ మెంబ్రెయిన్ కి దగ్గరలో ఒక విధంగా చెప్పండి అంచు భాగం దగ్గరగా ఉంటుంది అనమాట. బట్ ఇఫ్ యూ అబ్జర్వ్ యానిమల్ సెల్, దట్ ఈస్ వెరీ సెంటర్, అంటే అంటే సెల్ యొక్క మధ్య భాగంలో ఉంటుంది అనమాట. ఏంటది? న్యూక్లియస్. ఓకేనా? సో దట్ ఈస్ ద గ్రేట్ డిఫరెన్స్ బిట్వీన్ ప్లాంట్ సెల్ అండ్ యానిమల్ సెల్. ఆల్రెడీ ఐ టోల్డ్ వన్ మోర్ డిఫరెన్స్. వాట్ ఈస్ దట్? What is that? The plant cell, actually, this is the animal cell, this is plant cell. Both cells are having cell membrane. Means the outer covering of this cell. అంటే ఈ సెల్ దేని చేత కప్పబడి ఉంటుంది? రెండు సెల్స్ కూడా ప్లాస్మా మెంబ్రేన్ ఆ సెల్ మెంబ్రేన్ తో కప్పబడి ఉంటాయి. ఫర్ ఎగ్జాంపుల్, దిస్ ఈజ్ సెల్ మెంబ్రేన్. దిస్ ఈజ్ సెల్ మెంబ్రేన్. వాట్ ఈస్ దట్? This is our cell membrane. Both cells are covered with the cell membrane. Do you know? The extra layer we have to see on the plant cell. It means above the cell membrane, there is another structure we have to see in the plant cell. That is called cell wall. What is that? Cell wall. But the cell wall we can't see on the animal cell. Okay, na? If you see the inside the structure of the cells, inside the cells there is a fluid-like structure or fluid-like material. What is that? Fluid-like material. A fluid-like material. Inside the cell, there is a fluid-like material. This is called cytoplasm. What is that? Cytoplasm. What is this? Nucleus. Nucleus is an important part in the cell. It means an important part in the cell అని అన్నానంటే న్యూక్లియస్ తో పాటు ఇంకా వేరే పార్ట్స్ కూడా ఉంటాయి అన్నమాట. ఇట్ మీన్స్ 1, 2, 3, 4, 5, 6, 8, 9, 10. These are all the parts. Do you know what actually these are all the parts we have to see in the cytoplasm. What is this? Cytoplasm, a fluid-like structure. In the fluid-like structure, nucleus and also some other parts also is there. Do you know what is that? Cell organelles. What is that? Cell organelles. సెల్ ఆర్గనైజ్ అంటే ఏంటి? సెల్ని ఆర్గనైజేషన్ చేస్తాయి అన్నమాట, సెల్ని నడిపిస్తాయి అన్నమాట. దే ఆర్ కాల్డ్ సెల్ ఆర్గనైజ్. ఇన్ ద సెల్, nucleus and also some other parts, they are called, including nucleus, they are called cell organelles. Among these all the cell organelles, who is the main important cell organelle? That is called nucleus. Do you know one thing? Nucleus is the leader of all the other cell organelles. The nucleus is a controller of the cell. అంటే సెల్ ని కంట్రోల్ చేసేది ఎవరు అంటే న్యూక్లియస్ అన్నమాట. సెల్ ని కంట్రోల్ చేసేది ఎవరు? న్యూక్లియస్. These all the organelles work under control by nucleus. సో ఇంత ఇంపార్టెన్స్ ఉన్నటువంటి న్యూక్లియస్ ని కనిపెట్టింది ఎవర్రా అంటే దట్ ఈస్ ద సిగ్నిఫికెంట్ అబ్జర్వేషన్ ఆఫ్ రాబర్ట్ బ్రౌన్. హూ ఇస్ దట్ పర్సన్ నేమ్డ్? రాబర్ట్ బ్రౌన్. We have to learn about the cell organelles in next class, that is called 9th class. Okay, these all are called cell organelles. So these both are plant cell and animal cell. In the plant cell we have to see cell wall, but animal cell this is opposite. Inside the cell there is a liquid material that is called cytoplasm. A dot-like structure is there that is called nucleus, and also some other structures also is there. They are all called as cell organelles. We will learn about the cell organelles in the 9th class. Okay, thank you.
To prepare stained temporary mount of onion peel. Materials required: medium-sized onion, slide, cover slip, watch glass, forceps, knife, needle, distilled water, glycerine, dropper, blotting paper, saffron in solution, compound microscope, brush. Procedure: take a medium-sized onion and cut it laterally into 4 to 5 pieces with the help of a knife. Remove a scale leaf from one of the pieces. Break the scale leaf gently from the middle and peel a small portion from the delicate epidermis, that is the covering, that covers the inner surface of the scale leaf. Remove the delicate epidermal peel with the help of forceps and place it in a watch glass containing distilled water. Take another watch glass and place in it 3 to 4 drops of safranin solution. With the help of brush, transfer the peel in water into the safranin solution for staining. Wait for 2 to 3 minutes. Transfer the peel back to the watch glass containing water. This will remove the extra stain sticking to the peel. Take a clean glass slide and pour 1 to 2 drops of glycerine. Transfer the stained peel on the drops of glycerine on the slide with the help of brush. The peel may tend to curl up. With the help of needle and brush, flatten the peel. Place the cover slip on the slide in such a way that no air bubbles enter in it. Gently press the cover slip with needle to spread glycerine and peel properly. Remove the extra material surrounding the slide with the help of blotting paper. Examine the slide under compound microscope. Observation: there are large number of cells lying side by side with distinct cell walls. A largely stained nucleus is present in the periphery of cytoplasm. A lightly stained cytoplasm is the periphery of cell wall. A prominent vacuole is the center of each cell, surrounding by cytoplasm. Inference: the cells under observation are the plant cell. It because cell wall and large vacuoles are clearly seen.